CNA Flashcards study material. Welcome to the quiz. Question 1. During the propagation of an action potential along a myelinated axon, what is the term for the rapid, jumping conduction between nodes of Ranvier, and what is the key biophysical advantage it provides? Option A. Temporal summation. It allows for the integration of multiple subthreshold signals. Option B. Saltatory conduction. It increases conduction velocity and conserves ionic energy. Option C. Electrotonic conduction. It ensures the signal degrades over distance. Option D. Synaptic transmission. It allows communication with the next neuron. The answer is B. Myelinated axons are wrapped in a fatty insulating sheath called myelin, created by oligodendrocytes in CNS or Schwann cells in PNS. The myelin sheath is interrupted at regular intervals by small gaps called nodes of Ranvier. Voltage-gated sodium channels are densely clustered at these nodes. When an action potential is generated at one node, the depolarization spreads passively but quickly through the insulated internode segment to the next node. This passive spread is fast because the myelin prevents current leakage. The depolarization is strong enough to reach the threshold and trigger a new action potential at the next node. This process, where the action potential appears to jump from node to node, is called saltatory conduction, from the Latin saltar, to jump. The key advantages are speed and efficiency. Conduction velocity is much faster than in unmyelinated axons of the same diameter because the signal doesn't have to be regenerated continuously along the entire membrane. It also conserves energy because ionic currents and the subsequent work of the Na/K pump to restore gradients are needed only at the nodes, not along the entire length. Option A, temporal summation, is the addition of postsynaptic potentials over time at a synapse. Option C, electrotonic conduction, is the passive, decremental spread of a voltage change that weakens with distance. It is not the mechanism for long-distance signaling in axons. Option D, synaptic transmission, is the process at the end of an axon, not the mechanism of conduction along it. Question 2. At the chemical synapse, exocytosis of neurotransmitter is triggered. What specific role do snare proteins like synaptobrevin and SNAP25 play in this process, and what toxin can cleave them to cause paralysis? Option A. They act as receptors for the neurotransmitter on the postsynaptic membrane, curare. Option B. They are enzymes that synthesize acetylcholine, organophosphates. Option C. They are voltage-gated calcium channels, tetrodotoxin. Option D. They mediate the docking and fusion of synaptic vesicles with the presynaptic membrane, botulinum toxin. The answer is D. The release of neurotransmitter at a synapse is a highly precise form of exocytosis. Snare proteins are the core molecular machinery that makes this fusion happen. They're like specialized ropes and clasps. Synaptobrevin, also called VAMP, is on the synaptic vesicle membrane. Syntaxin and SNAP25 are on the presynaptic membrane, the target membrane. These three proteins coil around each other to form a tight snare complex. This complex pulls the vesicle membrane and the presynaptic membrane into close contact, overcoming the repulsion between them and catalyzing their fusion. This creates a pore through which neurotransmitter is released into the synaptic cleft. Botulinum toxin, produced by the bacterium Clostridium botulinum, is a protease enzyme. It works by entering nerve terminals and cleaving specific snare proteins. It has different types that target synaptobrevin, SNAP25, or syntaxin. By cutting these ropes, botulinum toxin prevents vesicle docking and fusion, blocking neurotransmitter release and causing flaccid paralysis. Option A. Receptors are on the postsynaptic membrane, snares are on the presynaptic side. Option B. Acetylcholine is synthesized by the enzyme choline acetyltransferase. Organophosphates inhibit acetylcholinesterase. Option C. Voltage-gated calcium channels let calcium in to trigger the process. Tetrodotoxin blocks voltage-gated sodium channels, not calcium channels or snares. Question 3. The blood-brain barrier BBB is highly selective. Which cellular structure, formed by specialized endothelial cells, is primarily responsible for its restrictive permeability, and what type of molecules generally cross it most freely? Option A. Tight junctions, small, lipid-soluble molecules and gases e.g., O2, CO2, ethanol. Option B. Gap junctions, large, hydrophilic proteins and antibodies. Option C. Astrocyte foot processes, ions like Na and K. Option D. The choroid plexus, glucose without specific transporters. The answer is A. The blood-brain barrier is a protective filtering system in the capillaries that supply the brain. Unlike capillaries in most of the body, brain capillary endothelial cells are fused together by very tight, continuous bands called tight junctions. These tight junctions seal the gaps between endothelial cells, preventing substances from leaking between cells' paracellular transport. This forces all materials to pass through the cell's transcellular transport, which is highly regulated. Because of this, the BBB is most permeable to substances that can easily diffuse across the lipid bilayer of the endothelial cell membranes. These are small, uncharged, lipid-soluble lipophilic molecules, such as oxygen O2, carbon dioxide CO2, and certain drugs like ethanol and caffeine. Larger molecules, charged ions like Na, K, and most water-soluble hydrophilic substances cannot cross unless they have specific transport proteins. Option B is incorrect. Gap junctions are channels that connect the cytoplasm of adjacent cells, allowing direct communication, and are not the primary barrier structure. Option C. Astrocyte foot processes surround the capillaries and help induce and maintain the BBB, but do not form the primary physical barrier, that is the endothelial tight junctions. Option D. The choroid plexus produces cerebrospinal fluid CSF and forms the blood-CSF barrier, which is different from the BBB. Glucose crosses the BBB via specific GLUT1 transporters, not freely. Question 4. In the visual pathway, damage to the optic chiasm specifically affects vision. Which pattern of visual field loss is characteristic of a lesion at the central body of the chiasm, compressing the crossing fibers? Option A. Complete blindness in the ipsilateral eye. Option B. Bitemporal hemianopia, loss of both temporal visual fields. Option C. Homonymous hemianopia, loss of the same half of the visual field in both eyes. Option D. Central scotoma with preserved peripheral vision. The answer is B. The optic chiasm is the X-shaped structure where the two optic nerves meet. Here, nerve fibers carrying information from the nasal inner half of each retina cross over to the opposite side of the brain. Fibers from the temporal outer half of each retina do not cross, they stay on the same side. A lesion at the central body of the chiasm, e.g., from a pituitary tumor pressing upward, damages these crossing fibers. Since the crossing fibers come from the nasal retinas and the nasal retina sees the temporal outer visual field, damaging them causes loss of the temporal visual field in both eyes. This is called bitemporal hemianopia. It's like losing the peripheral vision on both sides. Option A would result from complete damage to one optic nerve before the chiasm. Option C, homonymous hemianopia, e.g., loss of the left visual field in both eyes, results from damage to the optic tract, lateral geniculate nucleus, or visual cortex after the fibers have sorted at the chiasm. Option D, a central scotoma, is typically from damage to the macula or optic nerve head, not the chiasm. Question 5. Parkinson's disease involves degeneration of a specific dopaminergic pathway. Which nuclei in the midbrain are the primary source of these neurons, and what is the major clinical consequence of their loss on basal ganglia circuitry? Option A, locus coeruleus norepinephrine, results in intention tremor and ataxia. Option B, raphe nuclei serotonin, results in depression and sleep disturbances. Option C, substantia nigra pars compacta, results in increased inhibition of the thalamus and bradykinesia/rigidity. Option D, ventral tegmental area, results in a flattening of affect and anhedonia. The answer is C. Parkinson's disease is characterized by the progressive loss of dopamine-producing neurons in a specific region of the midbrain called the substantia nigra pars compacta SNC. These neurons project their axons to a part of the basal ganglia called the striatum caudate and putamen, forming the nigrostriatal pathway. In the normal basal ganglia circuit, dopamine from the SNC facilitates the direct pathway, which promotes movement, and inhibits the indirect pathway, which suppresses movement. The net effect is to allow desired movements to proceed. When dopamine is lost, the indirect pathway becomes overactive, and the direct pathway becomes underactive. This leads to excessive inhibitory output from the basal ganglia, specifically from the internal segment of the globus pallidus, GPI, to the thalamus. With the thalamus excessively inhibited, it cannot properly activate the motor cortex, resulting in the classic motor symptoms of Parkinson's: bradykinesia, slowness of movement, rigidity, resting tremor, and postural instability. Option A describes the locus coeruleus, which produces norepinephrine. Its degeneration is associated with conditions like Alzheimer's, but not primarily Parkinson's motor symptoms. Option B describes the raphe nuclei, which produce serotonin. Option D describes the ventral tegmental area VTA, which sends dopamine to limbic areas and is involved in reward and motivation. Its dysfunction is more linked to psychiatric symptoms, not Parkinson's primary motor deficits. Question 6. Cerebrospinal fluid CSF has several key functions. Where is the majority of CSF produced, and through what structures is it primarily reabsorbed into the venous system? Option A. Produced by the ependymal cells lining the ventricles, reabsorbed via the choroid plexus. Option B. Produced by filtration from cerebral capillaries, reabsorbed into lymphatics. Option C. Produced by the choroid plexus in the ventricles, reabsorbed via arachnoid granulations into the dural venous sinuses. Option D. Produced in the subarachnoid space, reabsorbed via the foramen of Magendie. The answer is C. Cerebrospinal fluid is a clear fluid that cushions the brain and spinal cord. The majority of CSF, about 80, is actively produced by specialized structures called the choroid plexus. The choroid plexus is a network of blood vessels and ependymal cells located within the ventricle's hollow spaces of the brain. It filters blood plasma and secretes CSF. CSF flows from the ventricles into the subarachnoid space surrounding the brain and spinal cord. It is not reabsorbed by the choroid plexus. Instead, CSF is primarily reabsorbed back into the bloodstream through structures called arachnoid granulations or villi. These are projections of the arachnoid mater, the middle meningeal layer that protrude into the dural venous sinuses, which are large blood-filled channels. The pressure of the CSF pushes it through one-way valves in these granulations and into the venous blood. Option A is incorrect because while ependymal cells line the ventricles, they are not the primary producers. The choroid plexus, which includes modified ependymal cells, is. Also, the choroid plexus produces, not reabsorbs, CSF. Option B is incorrect. CSF is not a simple filtrate from general capillaries, and lymphatic reabsorption is minor in the CNS. Option D is incorrect. The foramen of Magendie is an opening that allows CSF to flow out of the fourth ventricle into the subarachnoid space, not a site of reabsorption. Great job! Don't forget to subscribe. Bye!